Right. Hi, I'm going to show you how to do an RNA-seq analysis of Naegleria fowleri. This is a small little tutorial. So the first thing we're going to do is this paper. It's called Transcriptomic Profiling of Brain-Eating Amoeba, Naegleria fowleri. We're going to use from this paper. So this is the bio project number here, and then in NCBI we can search that. And then we are able to find like all of the runs that were ever done with that bio project right there. And so then we get all of the SRR runs and then all the information about them. In this tutorial, we're looking at the NF45 high virulent strain at day 5 versus the NF1 low virulent strain at day 5 and all the replicates that are associated with that. And so with that, there's 6 runs that we want to include. And so I've copied all of those SRR file names so that we can pull them. So we have this get data in the tools section of Galaxy. There's all sorts of different tools, but get data, the second one is download and extract reads in FASTQ format from SRA. So we want to do a list of SRA accession numbers, 1 per line. So what we're going to do, I've already kind of done this a couple times before, as you can see, but I want to make a list, a text file with all the different numbers that I want. So you put paste fetch data, and then we're going to call this SRR list. And it has all of my SRR numbers that I want. I'll press start. Perfect. And then I close. We can see right here when it's orange and like that means that it's loading and performing the operation for you. So it's going to create my little text SRR list here. Perfect. We can see our SRR list is now here. We click on it. We can look at different things we can do with this list. We can download it, we can copy the link, dataset details, visualize. This eye here is for view. We can see everything that's in there. Perfect. All right. The other thing that we need for this analysis is we need to have the genome so that we can map the reads of the SRR to it. Oh, but sorry. First, what we'll do is we'll take this download and extract reads, use the list of SRA accession 1 per line. It's going to include our little list here, and it's going to run. So now what it's going to do is it's going to pull all of the FASTQ files of all of those runs in here for us. Our data sets have paired end reads, so they're all going to show up here, not in the single end read. While that's going, this gray and like the clock means that it's kind of like in queue waiting to be processed because Galaxy, it's not like run on a server. It's not run like, locally. It's run basically on the cloud from my understanding. So the other thing that we're going to do that we need is we need to be able to map these reads to the genome. So we are going to find the NCBI datasets genome, download genome, sequence, annotation, metadata, da, 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 da. So this is the genome assembly that I will want to be using. So I'm going to copy this GCA number here, and then how to find the genome. I want it by assembly. I'm going to enter the accession number, and I'm going to enter that right there. Then what I want to include, it's going to load there for me. So it's going to send me the genome, which I want. I want the FASTA sequence that I can map to. But the other thing that we want is called the gene transfer format, which has all the annotation data. These are the 2 things that we need for this experiment, this tutorial. So then you'll press Run Tool. And it's again going to show up here, and all of these are waiting around. So after those are ready, we'll move on. All right. So as we can see here on the right-hand side in the history, we've got the paired end data from the tool that uses like this FASTQ dump. We've got all of these, and we've got our forward and reverse. And I've just clicked right here on this forward one, and we can see this looks like a normal FASTQ file. So great. That means that we've got, and then on the other hand, we have our genome, the FASTA file. It's loading. See if that works. We'll give it a sec to load. We've also got the GTF file right here. And so that way we have everything that we need. All right. So now that we have all of our data is in here, what we can do is run our workflow. And so this is the first workflow that does the trimming for us. So we'll input our forward and reverse sequences, and then Trim-O-Matic is going to be used here to be the paired end, and then we are going to use an Illumina clip step to remove the TrueSeq 3 adapters. Additionally, the RNA star is also set up to run with the paired ends. Actually, what we are going to do is the GTF file. We're gonna input that specifically, and it's gonna go for the RNA star and for feature counts. So then we've got RNA star, and that leads into feature counts, and then fast QC is run for Trim-O-Matic after trimming, and then multi-QC is run after RNA star and feature counts. To be able to, multi-QC just looks at basically how well was the tool run, the RNA star, the feature counts. And so we'll look at that in a little bit. So, and this workflow, when you design your workflows, you can use your input data sets. You can use all the different tools that are here, and you can input them and connect the outputs to each other, which is really nice. And I'm just going to press save and exit. So then I'm going to run the workflow. So what are we going to do? We are going to input dataset 1 and 2, the forwards and reverses, and then the GTF here. So we'll start with our multiple data sets. I'm going to go to the paired ends and get all of the forward reads. Select all of those. And then number 2, I will come in here and get all of the reverse reads. So it'll run like that workflow a lot of different times, and they'll be just like in different batches. And then with the GTF file, we're going to go under NCBI GTF, right, that we imported, and it'll be there. And then we're going to use the reference genome, which is that reference genome that we have right here. Again, the assembled one, right? And then everything else is set, ready to go. You can look at all the different options that you picked, and you can kind of edit them here. I like to edit them in the whole workflow though. And then at the very top, you will hit run workflow. So it's going to run this workflow 6 different times because, as we know, there are 6 different samples, and there's forward and reverse for both, so it's got to run the whole thing. And if you see going on here, it's just populating with all the different jobs that it's got to do, right? So for each of those, there are a lot of jobs. I will go to workflow invocations, and if we open that up, see, it's scheduling my workflows right now. So this is one of the workflows. It's doing one of the reads, the forward and reverse, and we can see that it's currently running the Trim-O-Matic function. And if we scroll down, we can see that some of these have already started doing the Trim-O-Matic. They don't want one person running too much all at the same time, so they'll kind of do it in so the Galaxy will do it in waves so that you're doing Trim-O-Matic on some of your samples, but not all of them right now. And then once the Trim-O-Matic is done, it will kind of start doing the fast QC and the RNA star just in process. the trimmomatic and all of this takes quite a while, as does, I think you probably figured out, the importing the paired end reads. That takes a while. So I will come back after this is done, and then the DE-seq step is actually really quick. It's the importing the paired end reads part, and then doing this, having this workflow run multiple times. Those are the steps that take a while. I sometimes leave this running overnight or for a couple hours while I work on something else, and we'll come back in a while and then do those next steps. All right, so now that we've finally finished this first workflow, we can see here everything is green, which is super nice to see. Again, remember we had to do this for 6 samples. So I'll go through a little bit about what these things did. So first we have our trim-o-matic that was able to trim off the adapters and the um, and yeah, and any very low quality bases. If we load this, it will be very, very long. The best way, you see, yeah, the data set is very large and it's just the FastQC or the FastQ file. So what they have is FastQC allows us to look at the quality of these different sequences now and the reads now post trimming. We'll see if that loads for us. So I am waiting for this to load. Great. So what we can see here is that for all the different bases, what is the quality? Most of them are above 30, which is great. There maybe might be a couple bases that are a little bit lower, but still pretty high. Then there's all of these other QC reports that you can look at and look up more. The biggest thing I'm looking for is this adapter content to make sure that there's no adapter still present. So that's what we're looking for there. So that's the fast QC, and you can see that on both the forward and reverse because that's what we've done. And then RNA star is going to do the mapping, and we've got our BAM file right here. But BAM files are not really fun for us to read. Computers like to read them, but not us. So what we do is, again, we have this multi-QC, which looks at the RNA star logs to say how well did we do. So it's going to load here. Takes a little while. Perfect. So here's our multi-QC. And what I like is this one here. We'll close it up. Perfect. So for this RNA star, we can see that 82.5% of the reads uniquely mapped to the genome. And then we've got some that mapped to multiple loci, some that didn't map because they were too short or for other reasons. And so that's all listed here, which is really nice to see how well did our sequences align. You know, if you're only getting like 20% aligning, you might need to look a little bit more at your sequences. So then the last thing in the workflow is the feature counts. And let's look at that counts for us. That should be generated. Loading, loading, loading. Perfect. And we can see here, these are all the different gene IDs of different genes. And then these are the counts for this specific data set, right? So then there are going to be 6 of these feature counts that we have. Okay. I just went through and labeled all the feature counts. I'm going to show you how to do that here in case you need to change the name of something. What you do is you hit the edit attributes and it will load, hopefully. And then you can change the name here. So I'm going to put NF45-3. So this is one of the NF45. It's the third replicate. So I'm going to save that. This is super helpful to be able to relabel them, especially for what's coming next. So I'm going to go to workflows. And I'm going to go to the my DESeq2 analysis. Now, to kind of walk you through what this looks like and what it's doing, we'll just wait for it to load. Perfect. Alrighty. So first we have our DESeq2, and I'm gonna open it up so we can see it a little bit more. So the factor that I'm looking at is virulence, right? And so there are two factor levels, like, this, as it says, it could be tumor, normal, treated, control, right? So I'm labeling one as NF1, and the other one is NF45. And then we'll put the counts files in here when we run the workflow. So, and then my choice of input is count data from something like feature counts. Perfect. And then I also selected this output, like I want to see all of the plots. So that's what's going on there. And then we're using that GTF file that we have to do the annotation. And so when the DESeq2 and then the annotation together, it will do this little annotation table for us, which is really nice. And then there's a little header that we need to add. So we'll go about adding that header, which reminds me I need to move that in here. And so then this tool will allow the header to be attached to the table that it's going to make, and then we're going to do two things with it. One, just make a volcano plot, and then the volcano plot I've set to it's going to label the top 20 significant DEGs. And then we're going to take this table and then we're going to filter it to have only genes that with adjusted p-values of less than 0.05. And then we're going to make two tables, one that is genes that are greater than 1 log2 fold change and then less than -1. So those will generate all the tables that we need. So we're gonna save and exit this. I'm gonna quickly get the header. The header, what you can do, I will show it to you once I bring it into our tutorial. Okay, I just imported the header because I didn't want to remake it. But this header just has all of the headings for the table that the DESeq2 plus the annotation is going to make. And so as we wait for that to load, perfect. So it's got the gene ID, log2 fold change, the p-value, adjusted p-value, the chromosome, it just has all this information here in a header. So, and I find that that's incredibly helpful. I got this from the tutorial for the RNAseq, so that's where this came from. So we're going to go back to workflows, and we're going to, again, run this DESeq2 analysis. So what we need to do, sometimes it just thinks that everything it wants is the header. So what we're going to do is we're going to provide all of the counts for the NF1. So what I do is I type in NF1, and because I've labeled all of them, which is not good. And then I just select all of them. Perfect. And then I'm going to do the same thing here, NF45, and get all of the counts tables here for the NF45. Perfect. And then the header labels. Again, remember this is the whole workflow, so I've added the header here. Then we're gonna provide that GTF file. So now we have so many data files. This is so great. here we go. So our NCBI, the GTF file right there. And then everything else is good to go. So we're going to hit the run workflow and see how it goes. See, this is the nice thing that I really like is once it loads, you can see the workflow and um, kind of see what steps it's on. Sometimes it will run multiple steps at a time. So, yep, we can see that it's running DESeq2 right now. It's already imported the other files because we already have them, and it's going to run DESeq2. And then when that's finished, we'll look at all of the results. So luckily this one's a lot shorter. It takes not even 5 minutes, I think, to do all the sequence. So that's super nice. Let's look at what we got. So we have our DESeq results file, right? That is going to have all of our information, right? The gene ID, the log fold change, the p-values. Nice thing is that they're sorted by adjusted p-value. Then we've got all of our plots right here. You know, things like our PCA plots and our dispersion estimates. Do histogram of p-values, our MA plots, all right here. And then a normalized counts file. So normalizing, it does all the normalization for us here in this file. So that's really nice. See if that loads for us. There we go. So for each of these genes, these are the normalized counts for all of them. So, yep, that's what we've got here. Then this is the table, like the annotated table that I was mentioning. So it has the genes and it has, again, like the log fold change and then where it is on the chromosome, all of that. And that's why we need to add the header because we kind of lose the header with that step. So we add the header back in here. Sorry, large data sets, you know what I mean? So, but still, Galaxy is able to handle them pretty well, which is nice. So now we've just added our header to the top here. And then we've got our filters, and we'll first look at the volcano plot. So here's our volcano plot. Yeah, you can see, you can adjust the volcano plot as you want to look at it a little bit more, look at it better. And then we've got our two tables here. This one, if you're like, oh, what in the world did I do to this table to get it? You can click on it and it says right here, filtering with C3, which is the log2 fold change greater than 1. Oh, so now I can see I've got 515 of these. These are all the genes that match that. And then here are if I look at this one, I've got all of the genes that have a log2 fold change less than -1. And it's kind of nice. It tells me how many of them how many we kept from the first filter, which I should say the first filter over here was the adjusted p-value filter. And then we further filtered it for the log2 fold change. So here we go. And then we have, again, what the gene ID is, the chromosome, start, end. Naegleria is a little bit tricky, right? Because it doesn't really have like the gene names or the features as much. And so what we did here is then we took these gene IDs and put them into David ontology to like the David gene ontology functional analysis to be able to see the different gene ontologies. And then again, for this volcano plot, what you can do is you can go to Uniprot, that's a pretty good source, and type in these IDs for the genes and specify that it's for Naegleria fowleri, and then you can figure out what all these genes do and what might be significant about them being up or down regulated in low or high virulent amoeba, brain-eating amoeba. Anyway, so that's the Galaxy workflow. And yeah, feel free to let me know if you have any questions.